at Novartis, and they took another approach. They just empirically screened, I think over 100 excipients against about 30 well-established tox targets that they have in these tox panels at Novartis and actually at many companies. And so this is a list of the excipients they looked at, and this is, activities that they saw across this panel. And wherever it's red, I mean, it's pretty potent. And so, for instance, this molecule, which is one of the dyes, one of the FD&C dyes, is this, 700, 800 nanomolar. I don't know if these are real-world data or not, but they are reproducible data. So there's a dose-response curve for this dye on the muscarinic receptor, and here's thimerosal. This is thimerosal. It's we were very cautious about even ever showing these data, but this is thimerosal. It's the molecule that has the anti-vaxxers all upset, and when we saw it, it made us upset, but for different reasons. It's about 100 nanomolar inhibitor of the D3, the dopamine 3 receptor. It's a metabolite. The thing it rapidly gets hydrolyzed to, ethyl mercury, is even more potent on the D3. They both get into the brain at higher than those concentrations. So this was an interesting, arguably scary observation, and you think so people when I heard thimerosal, I never knew that it was a mercury derivative, and you think, well, really, is this one of the things you think is, well, there's a lot of excipients are in our hands aren't active in anything, but in these days you wonder maybe we could do better than having a mercury derivative as an antimicrobial in our vaccines. Okay, so I think the takeaway from this talk is that actually, certainly at a molecular level, a substantial number, we don't claim to have done a comprehensive study, but a substantial number of the molecules that are defined as inactive ingredients are active fairly potently on molecular targets that are relevant for biology. And the second takeaway is that there's a systematic and comprehensive way to find what they are and then to explore what impacts that might have on drug behavior. Thanks very much. Oh, yeah, I'll take questions if there are any.
Well, Brian, can I just ask you to repeat it?
Mm-hmm.
I want to know, the pharmacokinetics of all these. Is that known that they actually get in and get to the brain?
Yeah, great question. So Kathy asked, what's the PK? Do they actually get exposed? For thimerosal, not through our work, but for published work, it does get into the brain of at least chimpanzees. At concentrations in this range, in the range of its IC50 values. Propyl gallate we did ourselves, with Lauzo's group, and that one gets hydrolyzed pretty quickly. Its concentration gets to about 5 nanomolar, and its T1/2 is short. So actually, except in cases where there's diseases of the gut, we suspect that its impact is probably pretty minor. But people do have diseases of the gut, so we don't know, and yeah.
This is dead. Everyone know? No. Carl Peck, NDA Partners. This is great research, great regulatory research. My question is, have any FDA drug labels been modified as a result of your findings?
So our findings aren't published yet, so no. They haven't, and I think it would be premature. Well, except with thimerosal maybe, but I think it would be premature to modify any drug labels. I think it's what it tells is a way forward, so you can start to do that.
So, how many of the companies that own the products or manufacture or sell the products have been informed about this, so that they can undertake the studies that would inform a modification in the drug label?
Yeah. No, we haven't informed anybody. We're just trying to get it published. So I would like you to write your. Okay.
Thank you, Brian. So we're going to move on to the panel discussion on advancing discovery to first-in-human clinical trials for new medical products, which will be moderated by Joe Wu and Jay Bradner. And I'd like to ask the panelists to come on up, Hal Barron, Tate Aldous, Matai Mammon, and Peter Marks.
So we're going to kick off this last session for the panelists. And I think you can pretty much look at our bioskets at the bioskets, so we're not going to spend time. I do want to reserve enough time at the end so that people can have a chance to ask the panelists questions. And so I'm going to serve more as a traditional moderator, and there are a lot of very, very smart boys and gals that are here who can answer all the questions that you have. So I'm going to just kick off by asking these 3 questions. And for each one of the questions, I'll ask the panelists probably 2 to 3 minutes. If you have more to say, maybe spend 3 minutes. If you have less to say, maybe 2 minutes. So we're going to go do this 3 times, and then afterwards I'll open this up for the audience here. So for the first question, it's listed there. In your opinion, which emerging capabilities promise to accelerate and improve the chance of success from discovery to first-in-human clinical trials? And for this one here, I'm going to ask my co-moderator, Jay, to start off. Jay, you have a lot of experience in this area, and I would say maybe some frustration as well. And I think you like to call this as patient segmentation, and that's a great way to phrase it. And I think the gist is that there are a lot of drugs out there for heart disease, COPD, asthma, lupus, and so forth, but we don't really have good clinical biomarkers or clinical efficacy tools to measure them. And what is the industry and also the FDA's perspective? How do you best measure clinical responses? So Jay, you want to kick? Yeah.
Well, thank you, Joe. I'm Jay Brenner from the Novartis Institutes for Biomedical Research. I'm a chemical biologist and a cancer doctor. Had the pleasure of training under Peter Marks a long time ago. This is one of our grandest challenges in drug hunting, is the ambiguity of human biology. And as I started to learn the discipline of ligand discovery and discovery chemistry, it seemed that the most insurmountable challenge was intrinsic disorder of target proteins, to fashion small molecules to achieve feats of biophysics and biochemistry only otherwise attainable by large molecules, but inside the nucleus. Now, fully calibrated to the capabilities of an extraordinary discovery engine at NIBR, I've found an even more humbling challenge is human biology. We have often great insights and leads from experiments of nature and human genetics, and where they're really clarifying, like PCSK9, they provide an absolute type of guidance you can get nowhere else. But as I adjudicate the governance board that receives new ideas from 5,600 dyed-in-the-wool drug hunters and diseases biologists, we spend most of the time in that meeting trying to piece through the line of hypothesis testing that we'll be undertaking to firm up our thesis in drug discovery. And this doesn't go away. After we declare a development candidate and enter human clinical investigation, the biology becomes even more complex because now we have the heterogeneity of the presentation of disease. As a leukemia doctor, we have absolute precision in how we define and subsegment patients by their cytogenetics, by their genotypes, by their clinical presentation, by the way the disease reveals itself to be responsive or not to therapy. And now I preside over an organization that makes medicines for asthma and COPD and bronchiectasis and Alzheimer's disease and dementias that may or may not be Alzheimer's disease. And I find our ability to understand and subsegment patients very poor and very humbling. And lacking this guidance, it's hard to imagine definitive medicines for these life-threatening diseases. So I don't know if this is the answer you were looking for when you crafted this. I don't think it's a regulatory challenge. I think it's a fundamental challenge in our ability to model and understand and iteratively test questions of human biology. Where we solve this, it opens the path for many important therapeutics. The lagging strand of pharmaceutical science is drug discovery. The challenging leading strand is human biology.
So thank you, Jay. That's a succinct 2 minutes, 20 seconds to the point. Okay, Hal, it's yours.
Okay, there's a challenge. I'll get my watch out. Hi, I'm Hal Barron, head of R&D at GSK, and I was going to say something similar to Jay, but I'll take it one step further. I agree that the biggest challenge is to find drugs for targets that ultimately will work for patients. And I think the question I would repose is not how do we do a better job of going from discovery to phase 1, because 90% of the things that get into phase 1 fail. So getting there faster is probably the wrong problem to try to solve. I would say that we should get things into phase 1 that have a significantly higher probability of success. And maybe that was what was intended by the question, but I'll interpret it that way. And with that as a problem, I think that the reason biology is so complicated is that we are a very, very complex species. And the biggest mistake I think we've made is to think that our understanding of biology is driven by understanding of mouse biology or fly biology or even potentially yeast biology. And that was convenient because understanding that in the human was impossible. But I think we now know that that system for identifying targets is flawed. And the preclinical models that we have in general don't translate into targets that work in humans. 90% failure rate, of which the vast majority is in phase 2, and that's usually due to lack of efficacy. So for me, the answer to both the heterogeneity problem that was posed by Jay, as well as finding better targets, is to actually use the human as the model organism. And to do so, of course, is challenging, but now with CRISPR and the ability to interrogate mammalian cells, and you probably heard a little bit about this in the prior session, but the idea now that we can use, we have massively large data sets with human genetics. There are quite a few more PCSK9s, I think, lurking. As the data sets get larger, we'll be able to see them when they're at a lower frequency. But the many, many thousands of polymorphic variants that we're seeing with the collaboration with Anne and 23andMe and other data sets like UK Biobank, FinGen, et cetera, don't have a clear gene product, as you mentioned, but we have, with the ability to do functional genomics, taking mammalian cells and doing gene-by-gene interaction maps, and being able to do now with precision editing, you can have isogenic controls where there's just one base pair change, and you can see the functional significance of one base pair out of 3 billion. And that will give us insights into a lot of human disease that will still be massively complicated, but I think choosing targets based on human genetics, functional genomics, and because these data sets in one experiment can be trillions of data points, particularly if the endophenotype that you're regressing on has got millions of endpoints like a transcript profile, this requires really very sophisticated machine learning to be able to unravel the semantic representation behind all this data. And when done, the human can be the model organism, we'll have better targets, and hopefully our success rate will go from, even if going from 10 to 20, which is going from horrible to not so good, will be transformational for the industry.
So thank you, Teja. Go ahead.
Hi, I'm Tate Aldous. I'm a chair of bioengineering here at UCSF, and I guess the only academic panelist today for this session. And I guess I have a slightly different perspective, and one of a researcher sort of sitting in basic science, really interested in how do we move our discoveries closer to human clinical trials, and what are the ways in which we can accelerate that. One of the reasons I came to UCSF was indeed, as an engineer, I could build a lot of technology, but that technology was sort of sitting in an environment where I was essentially decoupled from both the clinic as well as industry partners, sort of a traditional department of bioengineering in a large institution. And so, could we actually begin to think about technology from the standpoint of those that are really doing the translation in the industry side of things, but also the patient. And so as a researcher now, what has been really, I think, important to making that a reality is to, one, partner early with companies. So an example of that is sort of building on what Brian just talked about in his toxicity screening. which is to really think about, if we are creating medical devices, where are the material starting points that we can really leverage off of? We're not having a really huge chance of success if we come in with all new materials. But can we take existing materials and can we alter them from a functional standpoint and really do that early such that we can avoid immunological consequences? Two, can we leverage bioengineering technologies but really partner with those that are developing human mammalian cell culture, be able to do that to create models that are more representative of human biology. We know organ on a chip and other technologies exist, but I think the key, and we've heard this earlier, is to make sure we have quantitative metrics as to how those systems really correlate to the human biology. And then finally, from a standpoint of a researcher thinking about developing new technologies, really understanding the clinical ramifications and what those clinical studies are, and then working backwards. Because I think we're not taught that in any point in our education. And as we're now training that next generation of innovators, we really have to think about what is the actual roadmap to do that. And if we wait till it's too late, we're actually not going to be successful. So I'm actually putting together right now a training program for a bioengineering group, which is going in next week. And one of the key hallmarks of that is starting to get education around these issues first and foremost, not, okay, after I'm already got my PhD and I'm ready to go out and do something. It's how do we bring that in to understand how do we get to the clinic in a faster, more efficient way.
Thank you. Matai? Yeah.
Yeah, sure. So, the responses have been really great, so I'm trying to think of how to build on that. The job of an R&D organization is, one, to pick the right biology that if you engage it correctly, will make a meaningful difference to patients. So Hal and Jay have really addressed that one really nicely. The other two are to make the right therapeutic, the right molecule, the right protein, the right cell, make the right one, and that's not a trivial task. In some cases, that's not even possible. In other cases, it would take a very, very long time. And the other is to develop it in the right set of patients. Let me take that second one first, because that relates a little bit to the patient segmentation issue. And one of the things, that's just a fact at the moment about how medicine is practiced, how the regulatory agencies work, how most drug R&D organizations work, is that they categorize disease in a very particular way that's historical and anatomic. Oncology is kind of broken free of that to a degree and running trials called basket trials and other ways of segmenting patients, but most other areas of medicine haven't done that. So finding the right patients to develop your drugs comes down to viewing maybe the axes on which how patients are defined is not quite right. So it's not necessarily segmenting a disease that has a some name of it today, like finding different types of Crohn's disease, but rather it's finding what the response signatures might be to Crohn's and ulcerative colitis and psoriasis and psoriatic arthritis that are common in a way that signifies response to a particular biology, like IL-23, as an example, would be something we're interested in within Janssen. And if we could do that, then and the good news is there's as much lumping as splitting happening, and the group of patients that you put together are much more apt, if you've done your job, to respond to your molecule. So kind of the call to action in the medium term, it's not right around the corner, is to find appropriate response signatures for conditions that we hypothesize have a commonality to them given a certain biology. So finding the right patients for me is taking on that task. Making the right molecule, the most beautiful thing that's happened in, I think, the last 5 years in machine learning has to do with image analytics. And one of the things we're doing very, very deliberately and broadly within Janssen is using images of cells to basically look at parallel inference of biology. So instead of sequentially creating bespoke assays for a particular biology, we are looking at changes in image patterns as effectively the response, and creating dose-response curves using such images or other biosignatures. And doing that is much faster than creating a particular assay for every target that you want. And we've begun this process well before I came to the company, maybe 5 years ago, but we recognize it as having worked, and we're expanding it across the entire organization at the moment. That should hasten the creation of the right therapeutic that has the right degree of cleanliness and everything else you'd want in that therapeutic faster. So that's those are the two things that we're doing.
Thank you. Yeah, Peter, 2 to 3 minutes. Yeah.
Yeah, so I think I'll just echo whatever everyone else has said, but just try to put this in the perspective of rationally developed therapies. So when you think about gene therapies, and particularly about CRISPR-Cas9 gene editing, genome editing, we're going to need relevant models to deal with those products. And so there's obviously, we've been talking about efficacy models, but we need safety models. And it's already been the issue of organs on a chip, humanized mice, other ways of trying to get at what we'll need to get for safety information for these products, where we're going to have a lot of them coming through the pipeline. When you start to think about what you can do with genome editing and the need to actually figure out in a relevant model, which is something that's got human cells in it and a human genome, what the off-target toxicities might be. Having those models are something that are going to be really important as we move forward, because it's going to be safety that's going to, for that kind of rational drug development, which is going to be so important. So I think we're going to be seeing hopefully seeing some of the organoid models, which, despite the fact that they've been around for a little bit, they are not, many of them are not yet ready for prime time in terms of reproducibility. They're very great in any individual investigator's hands. I'm not saying that each individual investigator doesn't have them, doesn't have something that's working well. It's just that when you try to transfer them from one lab to another, they sometimes don't always transfer. So we need to have robust models there for determination of safety.
Okay, so we're going to move on to question number 2. And for the question number 2, for innovative new products, how should it says FDA, but I would also say, besides FDA, also industry and also academia. But here it says, how should the FDA acquire the scientific knowledge necessary to make good decisions about safety and efficacy? And for this one here, I would say we're going to move to the other extreme of the other end of the table by asking Peter first. So Peter, perhaps you can discuss an example, FDA's decision to recently criticize the gene therapy drug Zolgensma for treatment of spinal muscular atrophy by Novartis, and yet FDA was okay in terms of the clinical results and let the drug be on the market. So maybe you could perhaps explain that in the context of safety and efficacy. Yeah.
Yeah, so I think we need to pull that apart, which is that what I'll say is that, look, that if anyone out here wants to do forgery or fakery or stuff, you should just get out of the room right now, okay? Let's just not go there, okay? So that's all I'll say about that. Let's maybe confine ourselves to those, what should we deal with about decisions about safety and efficacy. I really don't want to think we want to talk about compliance. That is a compliance issue. Again, if you want to go fake, fool, just go out there. There are plenty of places you can go play 3-card Monte out here in the street for people in San Francisco, and maybe you can get people to put money down. When we're thinking about safety and efficacy, what we're really talking about, I think, are trying to have us as an organization have enough knowledge by being out there at meetings that are looking at the latest technology, at the latest genome editing technology, the latest drug modeling technologies, the latest AI technologies, and being out there and sopping that up. And our model at the Center for Biologics Evaluation Research is many of the people who do review work also do some type of research. And that, I think, is very helpful because they stay at the leading edge of the field. That actually, to me, is really helpful because the pace of progress here is becoming really almost dizzying. We're talking now about something like CRISPR-Cas9, which if this were 2011, we wouldn't be talking about because it hadn't been invented yet, and yet there's now an investigation of new drug applications in-house that have that technology. So it's a really exciting time here. I think it's a time that we have to, at the agency, really stay on top of our game of being knowledgeable. I think the CERSI program is really helpful to us in that regard. These kinds of involvements and staying involved with academic and industrial groups and staying on top of our game is really critical. And I'm sorry, I didn't mean to get so. The issue is that we don't like to talk about compliance and enforcement issues, but so that is kind of my way of just trying to deal with that.
Thank you, Peter. So, Mathai, you want to talk about industry's perspective on safety and efficacy?
So the question is, how do you acquire the scientific knowledge necessary to make good decisions? Is that the question? Because that's a weird question. How do you acquire the knowledge? Let me not answer that question and just say, how do you make decisions?
Okay,
To me, one of the interesting things on the first panel today that I heard is, don't be afraid of degrees of uncertainty and don't be afraid of non-traditional data sources. So to me, that's a really important point in that the randomized control trial, as we do it today and construct it, is an excellent source of data, and it has a certain degree of reliability associated with it. We make sure of that, we construct it that way. But there are lots of other data sources out there that one can use to augment the confidence that you have in whatever decision you're making, whether it be efficacy or safety. So to me, you don't need to go all the way to a synthetic control arm, or you don't need to do that fully. But for instance, we and all companies make good use of really amazing products that get a breakthrough designation. So when you submit a file that has a limited number of patients like that, the challenge around confidence around the safety and efficacy is particularly high, even higher than when you go through the normal full development program. So there, I would advocate for extensive use of other data sources. So so-called real-world data, potentially patient registries, potentially other natural history studies that have been ongoing. Anywhere you can get data, it'll never be like it is, and this is the comment about don't be afraid of uncertainty, but rather embrace it and account for it. Use those data in the most appropriate way you can to augment the conclusions that you're making. Maybe let me just stop there, and we can come back to that maybe in the context of the third question on how to do that.
Thank you. So, Teja, maybe 2 minutes. Yeah.
Sure. So I will go back to the question about acquiring knowledge. As chair of Kathy, I would say, CERSI is one of those mechanisms by which to really do that, and I really appreciate the partnership. If you had asked me 6 years ago who would care about regulatory science here at UCSF, I think there would be very few, but what CERSI has enabled is us to really think about how important the science is to actually getting to our end goal, and to really engage across disciplines, across departments and schools here, a set of expertise and the knowledge to really help to think about what are the problems and what are the solutions in terms of these new both biological therapies and also medical devices. So I think the FDA is aware of it, but then I would also add, I think the key to sort of completing the circle is to make sure that industry is involved in that same consortium. So it's not just FDA and academics, but it's really FDA, academics, and industry, and to make sure that support to find the very best people who are working in these areas, and then leverage that to be applied to informing the FDA.
So thank you, Hal. couple minutes.
Yeah, I don't have a whole lot to say because I don't think for the question for innovative new products, how should the FDA acquire scientific knowledge necessary to make good decisions about safety and efficacy? I think the FDA makes really good decisions almost always about safety and efficacy. So whatever the FDA is doing, I think is probably acquiring enough knowledge to do that well. The places where I think there could be room for improvement, potentially, and I don't know it's necessarily about knowledge, but it's just about the need for perfection or the willingness to accept ambiguity. I see areas like surrogate endpoints, whether it's pathologic CR, minimal residual disease, places where it's not perfect, but it's pretty good, and the advances that will be made by accepting those in terms of the impact for patients, if you take a longer view, will be, I think, worth the risk. And so I think it isn't knowledge so much, or you could say the same thing about safety. You know, we have now so much data on human knockouts for various genes. When we have 5,000 humans whose gene's been knocked out, we should think about that as a safety database. And when you're using an antibody to knock out the protein for 6 months, when there's 5,000 humans who've had it for life and they're fine, I'm not saying you shouldn't do safety, but at least it's another piece of the puzzle. And it's just pushing the envelope to say it's not perfect. There's definitely, you know, lots of ways these could be less than perfect, but the impact, if we're to embrace them, could be faster and more robust drug approval. So a little off question, but I think the FDA does a really good job of making good decisions on safety and efficacy.
Yeah, it's hard to add constructively after it's been pretty well considered. I'd have no instructive guidance to the FDA on how to learn. I think that it's pretty evident that that's happening. I can say that at NIBR, we've entered some areas that were very new for us. The scientific program I lead is in targeted protein degradation. The preclinical toxicology models that we have today just cannot inform on on-target toxicology. The cereblon ubiquitin ligase that we leverage, the isoform in mice, in rats, and in dogs is just incompatible with our chemistry. And so we've had to recreate certain preclinical safety paradigms. The same was true when we entered gene therapy. Read, listen, connect, recruit, tinker. And we did all of those things for years until ultimately we would acquire Avexis, which had cultural challenges, significant challenges in scientific misconduct, but an extraordinary capability to bring medicines to patients around the world, solving a drug access problem, having a field that had solved already discovery challenges, patient stratification challenges, pharmacology challenges, biodistribution challenges, payload challenges, genome structure challenges. We now had a drug access problem that they ultimately would solve for us. Cell and gene therapy and CAR T-cell, just incredible learning at a time when we thought that we were the experts. We learned quite humbly how hard it is to bring this medicine to patients all around the world. The race wasn't to proof of concept. The race wasn't even to FDA approval. It's still the race to address the burden of diffuse large B-cell lymphoma and B-cell acute lymphoblastic leukemia, overcoming significant manufacturing and quality challenges. The FDA is in a really unique situation, right? Because, Peter, you and your colleagues have access to data sets that are proprietary and incredibly granular and incredibly informative that really no one else in the world has a capacity then to learn through that narrative, through these cases and stories, but also has a challenge. And I can imagine working at the FDA that, that you recognize when we all visit, it's a big day for us. And we show up with all the data and knowledge and information we have, but it's a big and singular day, and innovation doesn't work that way. It's an everyday tinkering and looking around corners for what the next technology is. Just in point of fact, I've never visited the FDA. And so I'm certain that many members of my leadership team who think about the next generation of AI as applied to preclinical safety studies, could you do 7-day AI-enabled massively parallel nucleic acid detection tox and not have to do 60-day tox? Man, this would expedite the number of medicines we could bring forward for patients. And so I do think there is an opportunity on the connect axis to do better. And organizations like CIRSE, I think, could be really powerful organizations for this. And I say could be because it doesn't really happen in panel discussions. It happens in topical considerations where we just go to the depth of a new area to ultimately try to find the path forward to patients around the world.
So So thank you, and I do want to thank Peter and Jay for being so willing and open to discuss about the issues with the Avexis data set. So I'm going to move on to question number 3, which is how can key stakeholders play a role in supporting regulatory science outside the context of a specific product. And here I would say that the key stakeholders include not just sponsors of clinical trials or drug companies, but also regulatory agencies like the NIH or even the EMA, public agencies like the NIH, not-for-profit organizations like Bill and Melinda Gates Foundation or the Wellcome Trust, and also clinical trialists, investigators. Some of you are in this room. Patients, many of you are in this room, or healthy volunteers. Academia, journals like JAMA. You heard Howard talk about this this morning. And so for this one, I'm going to go back to Jay and have him start because he wore several hats. He was in academia, did clinical trials before he went to Novartis. So Jay.
No, thanks. I'll be brief here. I said a few comments to this thesis before. So I think that the FDA and the biopharmaceutical industry benefit when there is a Yenta, somebody who can bring us together around a topic, a new area, and we can enter into a more open and brainstorming and creative conversation around new and disruptive technologies that could accelerate discovery to first in human, for sure. I have a chance in my role to attend the IMI meetings, the Innovative Medicines Institutes Initiative in Europe. This is a great convener of creative thinking around new regulatory paradigms. Can we somehow get rid of the 6-minute walk test? It's insane to me. I get it, it kind of works, but it's insane to me that we're collecting more data on my wife's wristwatch right now, than in a regulatory endpoint for a new medicine that could affect each and every one of heart failure, severe respiratory disease, musculoskeletal disease, and certain neurologic diseases. We can surely do better, but we haven't, and that's with a capital W. This isn't an FDA issue, but it is something that I think that we can solve, and we're working together within one such IMI initiative to try to bring, the hybridization of accelerometry, machine learning, where appropriate, global positioning, to try to ultimately understand what it means to quantify humanity in the movement dimension. HEVER is another organization that brings top leaders together, but it's often not top leaders in an industry that really change and innovate concrete solutions. And so there are some Yentas out there that bring us all together, but I think much, much more could be done in this space. And my own leaning is that a lot of times we take on these big and kind of lofty conceptual conversations, but we never get deep enough to actually solve anything. And here I think organizing around definitive use cases, whether it's your tech or my tech, just having the series of cases where we've approached a complex use case and found a solution, perhaps like the ApoE example of 23andMe, that then charts a path for others that would dare to go down that road. I think if we can get very granular around use cases and have the right people at the table, which is not often the leaders, we can make real progress.
Yeah, I agree with all that. Let me just, I think one of the best collaborations I've seen that involve government, industry, and academia and patients, the 4 key components to anything that's going to be successful and transforming, has been Laura's iSpy. And I think what that provided was, what I've learned from that is that these terrific collaborations have certain things in common. There's a problem that everyone agrees is really important to solve, and that it's not product specific, it's for all companies and all clinicians and all patients who can benefit from faster, more thoughtful approvals. And that everyone has deep mutual respect in the 4 groups. It isn't driven by one academic, one government, or one industry player. It's something that each of the leaders believes the other groups have a lot to contribute and deep respect for them. And when you do that, it actually is not easy, but it goes well, and it makes advances. So I think the key thing is identifying people who care about the right things and are really deeply respectful of the other groups that are needed for that to happen. And when you do that, terrific things happen. I think the converse is when it's academic idea, it's an industry idea, it's a government idea, and they try to get everyone to buy into it as if they would support it, but it's not really embraced, and then everyone sits at the table pretending to be interested when they're not. I think if you really can find something that everyone deeply cares about, and it's not somebody's agenda, but it's what's best for everyone, hey, Ruta, then I think they work. So I think iSpy is a terrific example of how to do it right.
Thank you.
So, not too much more to add. I completely agree. I think, the way I think about it is really framing it around some of the grand challenges that we have as a community, and particularly in healthcare. And, exactly what Hal said is bring the people who care passionately about that challenge, as opposed to a singular product, a research project. No one's going to care about my individual NIH project, but really, what is the bigger goal that we're after? I also want to echo Jay in terms of bringing the actual people who are doing the work to the conversation, whether that's the students or the scientists or those who are engaging the patients. I think, again, it's happening at a panel or happening at some meetings. It's one thing, but really engaging the people who are going to make it happen down the road. Because these are not things that happen over one meeting or even a year. They happen long-term, and they happen in a multifaceted way.
Good. Matai?
For this one, I think I'd like to give an answer that has this layer of pragmatism just to it for a moment, and it'll conclude, I'll say some things, and it'll conclude in a task for all of us. I think that it'd be good to do. So, there's been so much talk of use of real-world data in clinical trials, so much talk of that, but there are a couple of very real problems with most real-world data. Problem number one is that the endpoints, and this comes to some of what we're talking about, the endpoints that we've constructed somehow over the decades in clinical trials can be awfully particular and far away from anything that a patient could possibly experience going to a doctor. And so there's different extents to which that's true. Sometimes it's exactly the same. It might be dying, or it might be a stroke, or it might be something like that, which is certainly measured in real life and in a trial. And it can go all the way to other extremes. The 6-minute walk is kind of a classic one that people complain about a lot, but there are many that are there at that extreme or somewhere along that continuum of delta in endpoints. So the delta between what is happening to a patient in the real world and what happens in measurements in a clinical trial. So I'll come back to that, but that's problem number one, is that delta. The other problem is adherence, and people don't talk a lot about that, but in the real world, there isn't perfect adherence to your medication. And so it's difficult to use a comparator from the world where adherence is, say, 50%, making it up, to a clinical trial where the adherence is close to 100%. So you can imagine, in a sense, like a 2x2, where you have high adherence or low adherence. Delta is close to 0, meaning it's the same, or delta is really high. So we're trying within our company to pick the quadrant where the adherence is high and where the delta is low, where it's almost the same. And then, as an industry, in order to get out of that little box, there's a task. And the task is to start to validate things that you can do in a clinical trial, working with FDA, working with the universities, so that you can creep slowly towards a world where you don't do these really odd measurements to patients, and you're freely drawing from, with confidence, the data that you can find in the real world. And so this is a big task, though. We can just talk about 6-minute walk as an example. We can talk about the information that can be drawn from EKGs as another example. EKGs, for instance, have all sorts of information in them that might correlate to things that we would otherwise measure in different ways. We're interested in our company in pulmonary hypertension, and we have a hypothesis that you can draw out of an analytics approach to EKGs enough information to maybe predict what the pressure is in your pulmonary artery as a hypothesis. So you don't need to necessarily go through all this work and expose a poor patient to sometimes it could be threatening kind of interventions to measure something that might be drawn from their experience in the real world. So our task is this constant, not product specific, so to get to the question, it's outside the context of a particular product, task is get together and start validating things in the real world that predict things that we would otherwise do in a trial.
So thank you, Matthias. So Peter, a couple of minutes before we open up to the audience.
Yeah, no, I think what everyone said, it's not very little to add. I think we at FDA need to continue to engage with stakeholders in kind of a robust dialogue of where the leading edge of science is in order to stay on top of our game. Ultimately, it's our job to kind of see through the haze of a lot of different competing interests and a lot of different competing information in order to try to find a path forward towards determining whether products are safe and effective to support their approval and in support of public health. And I think that's part of actually what all of this is about, is trying to use the best science in support of public health. And it's actually what helps us in some ways swim our way through problems when you do have issues of data integrity. At the end of the day, do we find is the essence of safety and effectiveness still there underneath all the noise that might be around that? And that's the kind of work that we have to do. But it's the foundational part is understanding the science that's behind it, and then if we have that foundationally, then it's easier to sort through all the noise that can happen when we see other issues that come up around the science.
Good. So we're going to open this up for the audience. We got about 15 minutes, and while the audience is coming up, so let me just start out by asking, I think Jay and Mathai made some criticisms about the 6-minute walk test. I'm a cardiologist, and so, it's not perfect, but there's some issues with the 6-minute walk test. But I would say that the alternative, there are a lot of discussions about wearable devices. I'll give you my personal anecdote. I got a Fitbit last Christmas. I wore it for 2 weeks, and I gave it to my son. He wore it for 2 weeks. He gave it to his younger sister. She wore it for 2 weeks, and I was sitting somewhere. We can't even find it. So in the first case, for the 6-minute walk test, at least Mr. Wu showed up for the 6-minute walk test before and after drug. The second case, how do you know it's Mr. Wu or somebody else? Yeah. So,
Wait, I have a quick anecdote.
Since the two of you make criticism about our 6-minute walk test.
I found out the other day that, by the way, that a 30-minute spin cycle on your dryer with a Fitbit, you walk 2 miles. Just remember that.
Go ahead, guys. Go ahead, Mildred. So I know the audience like to pontificate, but I would emphasize one person, one question. No pontifications. Go straight to the question. Yeah.
Okay, I can't top that. So Mildred Cho at the Stanford Center for Biomedical Ethics, and also I'm going to ask a question about this second point about acquiring knowledge to make good decisions about safety and efficacy from the point of view of someone who was a member of the now defunct Recombinant DNA Advisory Committee, and that's because we were asked to review, or we didn't really have review authority, but we were asked to advise on the early gene transfer trials that were first in human, and did that, and we're trying to advise those investigators who had proposed these studies, but all of us being in the dark about what other people's experiences had been with those kinds of new vectors and innovative interventions. So my question is, and it was very disturbing to sort of look at these CAR-T, very innovative CAR-T proposals and so forth, and then go home and then read in the paper that there had been deaths on other clinical protocols that were very similar, and we were not privy to that information, and neither were the investigators who were designing those trials that were being reviewed by the FDA. So I'm just wondering if there are any ways to make that kind of information available, especially for these new innovative mechanisms for his first-in-human studies?
Yeah, so it's a great question. The question here is why did the RAC, as it was constituted, the Recombinant DNA Advisory Committee, get transitioned? And actually, when the RAC was first brought forward to deal with issues on recombinant DNA, it was early on in the time of gene therapy. FDA did not have a tremendous amount of expertise in this area, but over the decades to two that since that the RAC was developed, FDA started to recruit and retain a number of very good scientists in this area who are able to now bring the knowledge, their knowledge of these products to their review. And they do have the benefit of seeing the entire portfolio. I mean, Jay already alluded to it. They see what's going on with the entire portfolio of CAR T cells or genome editing or whatever the technology is. So I think we internally can try to make sure that we protect the public to the extent that we can by, as we review things, making sure that we tell sponsors, we raise concerns that obviously we can't disclose what's in someone else's investigational new drug application. What's in an investigational new drug application is confidential, commercial, trade secret information. We don't share that. But that doesn't mean that if somebody got into trouble because of a certain construct, we can't tell someone, you need to go back and look at this construct. And that's the type of thing we do. Would it be nicer if it was a more transparent process? Maybe in some ways it would, but if there's this balance, and this is what we're constantly doing at FDA, this balance of wanting to protect innovators and wanting to protect the public, because ultimately you want the public to benefit from innovation. So that's the best I can answer that.
We're going to hit our questions. So Carl, go ahead. Yeah.
Carl Peck, I don't think I heard any mention of systems pharmacology in this panel. So it seems so obvious it's beginning to be applied for small molecules and other programs. Can you just comment on that, perhaps, Tejal?
As you know, I'm a strong supporter of a systems pharmacology approach, and I think one of the things that we've recognized and we've heard this throughout the day is sort of looking just at the sort of individual cellular level is not going to necessarily be predictive of the whole effect organismally. We were actually losing, I think, the sort of art of systems pharmacology, and to bring that back and as a sort of core tenet of how we think about drug-drug interactions, drug physiology, but again, with a really quantitative underpinning, is going, in my mind, going to be key to actually answering maybe question number one, which is how can we leverage that knowledge to really help with the acceleration to clinical application.
I don't consider myself a Yenta, I guess, but a question in that spirit. Really wondering if you could comment on the role of synthetic biology, specifically DNA writing, in functional genomics and that sort of translation from bench to bedside. I've been fascinated by as much as gene editing has enabled us to interrogate biology on a sort of a nucleotide-based level. What the next few years could possibly bring in terms of being able to rewrite very long pieces of DNA, as some professors, some scientists have been able to demonstrate, and recapitulate biology on a scale that's just been unachievable until now, whether it's complex regulatory sequences, long non-coding RNAs, et cetera. There's some intuition that there's a real there in terms of breaking through some of the complexity of the biology that we just don't get through mouse models, and wondering if that intuition is right.
Well, maybe I could start and then others could chime in. I'll take the liberty, as you did in your question, of considering synthetic biology as also including whole-scale and partial genome engineering. There's no question that the ability to synthesize DNA at great length and in a massively parallel fashion allows us an unprecedented view at variomics, the variation, engineered variation of a human allele in a disease biology or in a pharmacology setting. And we exploit this technology just every single week at NIBR, trying to anticipate and understand mechanisms of drug resistance, trying to identify the critical residues on a protein to then drive molecular creation. And so there's one aspect of synthetic biology that I think is surely well utilized by all at the table. You ratchet the bar a little bit higher and put it in a therapeutic context. And there it's been, I'd say, slow to catch on. True synthetic biology and being able to engineer whole organisms has been, at present, limited to prokaryotes, so others are working on higher systems. And the great challenge has been that your options have been to deliver an episome with AAV, suffer the random insertion problem with a lentivirus, which still as a leukemia doctor gives me chills, or to edit a single base or to vandalize a few bases. The ability to do true genome engineering has been limited by this holy grail of homologous recombination, as yet not penetrating high-efficiency therapeutic platforms. There are some signs that clades of viruses exhibit higher degrees of homologous recombination. There are signs from David Liu's lab that prime editing could allow one to insert larger sequences and potentially eventually get to homologous recombination events. But until we're able to directionally, safely, and purely replace and augment regions of the human genome, there are significant safety challenges with doing that in a therapeutic context. With that said, I think we're within a couple of years away from really seeing elegant therapeutic applications of that type of genome engineering.
So Jay, thank you for the eloquent answer. We're going to move on. Go ahead.
So as you've been talking quite a bit about the potential of genetics, either Mendelian genetics or complex genetic disorders and association studies to identify potential targets. And there are things that we can do as geneticists to improve the reliability and reproducibility of those associations in terms of replication, as well as Mendelian randomization technology to establish a greater sense of causality before we then move on into clinical trials. And I think that needs to be considered. The integration of omics, and you mentioned a number of them, with the genetics as well, is another means by which we could potentially establish means by which we could look at then multiple therapies. For example, in my field where retinal dystrophies are caused by more than over 120 or 150 different genes, we could then have commonalities that would allow us to implement therapies for more than one gene that causes a similar disease outcome. So I would be curious to know, and I asked this to the panel, to what extent do you see the possibility of actually broadening the criteria by which clinical trials are studied so that you could look at multiple therapies, essentially like gene-specific therapies for a single disease group at a time?
Yeah, let me take that. First of all, I think those are extremely well said points, and using quantitative trait loci or Mendelian randomization, things that give you a little bit better understanding of what's in the causal pathway and that what might be happening downstream from the gene are critical to making all this work. But I would add, again, the ability to do gene-by-gene interaction maps in a mammalian cell, particularly when you know the cell type of interest, is profound and brand new. And cheap and fast. And so marrying those two concepts with the ability to do this in mammalian cells, if you can find the endophenotype, which in what you were talking about, it's maybe in the eyes gives a lot of promise. And there were from that be a lot of opportunities to do gene-gene interaction maps, looking for suppressor genes. I mean, a lot of people have autosomal recessive diseases with incomplete penetrance. They probably have a modifier gene. And one could imagine that diseases like Parkinson's that has 5 or 6 different causal, almost assuredly causal genes in what appears to be very, very different pathways may converge. And so getting back to unraveling this deep semantic representation behind all these can really be done by gene-gene and gene-drug interaction maps that when you develop these trillions and trillions of data points in these data sets, the machines can start actually seeing things that none of us could see. And so they might start explaining why, example from my life, why rituximab, a developed drug for lymphoma, is a terrific MS and RA drug. It gets back to what Matai was saying. Machine learning can see these relationships that will allow us to think about drugs and genes for different diseases that might have, another great example is when you think of Gaucher's is to Parkinson's, where you see the autosomal form creating a disease in children that doesn't look anything like the heterozygous carrier form that ends up being one of the more common genetic drivers for Parkinson's. So these things are really related, but as clinicians, we examine things that are in the left side of the chest, and you become a cardiologist, or the back side and you're a pulmonologist. If we did it by biology and using genetics and functional genomics and all the techniques that you just described, we'd be able to see lots of relationships and therefore develop drugs and therapies in a much more robust way.
So thank you. Short question, short answer.
Laura Ventveer, UCSF. In my daily life, I'm actually responsible for the biomarker work in the I-SPY trial, which was just mentioned. And over the years, we have tested 10 different drugs and matched those with biomarkers that we called qualifying biomarkers based on mechanism of action in the literature. And actually I have a question for the whole panel. I think it was mentioned that you need to have like all the stakeholders at the table to do that in an efficient way. And both, I think, for this advancing discovery as well as for advancing clinical trials, I think a panel like you could be instrumental in really setting the framework how that needs to be done. So there's sort of many one-off discussions between pharma and academics, but there's not sort of like from the end what is known from all of the preclinical data till what is actually needed for the regulatory path to make it a companion diagnostics. To make sort of an efficient biomarker companion diagnostics. I think there is a lot to do, and the pathway that we've created in I-SPY with like a rigorous statistical framework is sort of helpful to move that. So I hope you would want to partner with us.
So, Matai, you want to take that, maybe the short answer, then the question?
So the very short answer is, I-SPY to me is like the original, broad-based, data-based decision-making framework. And so, the sort of instruction to all other people doing related things, that should be part of the mandate of I-SPY is to teach.
No, it is there, but still we're stuck to get it actually to the delivery.
Yeah. Last question. Yeah.
Thanks. It seems like so many of the excellent panelists' interest in regulatory science innovation has come about from trying to transition from one stakeholder to another, as in to try to develop a product. And so I'm wondering if anyone wanted to comment on the idea of having more first-in-human studies happen in academia, like lowering this walls between the different stakeholders where academia hands off to industry and then industry works with government, but if we could all kind of do that work together, if that would help advance discoveries.
So Hal, you want to answer this? Hal or Jay?
Yeah, I can. I think that there's an enormous amount of industry, academia phase one studies. There's academic industry phase one units. There's places I think that phase one is even better handled than academia. And I think for lots of reasons, I think cell therapy is an interesting example of where a lot can be accomplished in phase one through academic centers, and there's some regulatory reasons for that as well as scientific. But I think from where I sit and what I've experienced, the collaborations in phase one are actually, to some extent, more robust than any other phase where, because it's a very small number of academic centers because of its nature in phase one, and the collaborations are very intense. They're with very special places where phase ones can be done well, efficiently, because of course the industry concern is always that if the sites do anything even marginally inappropriate, you could identify a safety signal pretty quickly that's not drug related but trial related, and that gets people nervous. But choosing those outstanding phase one type partners is very common and very robust.
Okay, with that, we're going to bring this panel to a close. Thanks to all of the panelists for your insights. I hope what came through is there's quite a lot of work still to accelerate ideas from discovery to human clinical investigation, but hopefully you can tell from the tenor of all here and all of you in the room the true willingness to apply next generation technologies to make this even better for the patients we serve. Thank you all.
So before we go to George's introduction of Peter, I just want to remind everybody that we have a reception at 4 o'clock immediately following the program. It's less than a 6-minute walk. It's right outside this door. So please don't go anywhere. It'll be a great chance to ask those questions you weren't able to ask during the meeting.
Okay, we come to the last session of the day. For me, this has been a remarkable day. It was a great day. First of all, the topic's incredibly interesting, but probably like many of you, I've been to many meetings which consist of panels, and usually at least one of them is a real snoozer, and somebody goes on. In this case, I think every one of the panels was really extraordinary. So thank you for all the panelists and the audience participation. It's my pleasure now to introduce Peter Marks, who's director of the Center for Biologics Evaluation and Research at the FDA. This is a particularly interesting part of the FDA, at least to me. As you know, there's been an explosion of work in genetic therapies and cell therapies, and I think pretty much an astounding explosion in the number of applications that Peter's probably dealing with. And so these bring not only great promise, but I think as was alluded to in the last panel, great potential risks as well. And so tough balance, and the issues that are normally faced by the FDA, I think, are only heightened on this division. So without further ado, Peter Marks.
So thanks very much for hanging in there for a long day. I'm going to try to be relatively quick in my remarks, but this is going to be a little bit different, and now for something not completely different. I hope that by the end you'll see that a lot of this threads together. But one of the things we're very interested in doing is starting to think about how are we going to advance individualized therapies, which we have just decided to call bespoke therapies, because people, when they think about bespoke, first thing they have to do is they have to go to the dictionary and find out what bespoke means, if you're like about half the people I know. Then once me, who had to look it up, then you think, this really does make sense, and I'll try to convince you of that. But this is a time where I think we at FDA, with a lot of input from others, have been able to, I think, get out a little bit ahead of where things are going, and I'll try to show you that. Right now, we're in a period where we often talk about personalized medicine. And I think when we think about personalized medicine, we're talking about finding the right drug on the shelf to treat the patient. So the idea of personalized medicine is I get someone's tumor, analyze it, and look for what drugs we have on the shelf that might help that person's tumor be treated, eliminated. On the other hand, there's this concept now, I think, increasingly of individualized medicine, where we are creating the right drug to treat the patient. When you think about this, that is what ultimately we're doing with gene therapies, and what many might think is the natural progression of where we might go with genome editing technology, where there are probably, some of these are almost private mutations that lead to disease, and some may be mutations only in a few dozen individuals globally. So this issue here, though, is a very different paradigm than the one we're used to. Personalized medicine, the idea that you have a bunch of things on that you can do trials, you have approved products, and then you take them off the shelf. But individualized medicine means you're now having products which are going to be created for very small populations. We're not used to approving products for very small populations. That's not to say that we have not at FDA done so, because it was just mentioned, someone kindly mentioned, I think it was Hal might have mentioned Gaucher's disease. Gaucher's is a great example of something where the main trial that led to that approval was only 12 patients large. Now, there were other patients treated in the safety data set, but that was the product for that at the time was Ceridase, was something, and that was back in 1990. So this has been going on for some time. So this isn't totally new, but it's a different order of magnitude here, because now we're talking about many products that will have small amounts or small numbers of patients treated. And really, what's the importance of therapies for these disorders that are very rare? If you think about it, out of thousands of rare hereditary and acquired diseases, there are hundreds of disorders that affect 1 to a few dozen people per year that could be addressed by novel therapies. That includes antisense oligonucleotides, phage therapies, and cell and gene therapies, including genome editing. And if you think about it, when you start to take apart some of the more common uncommon diseases, you start to realize that if we address those with genome editing, where you start to think about making constructs that might address any one of between 10 or even hundreds of mutations that lead to a given disease phenotype, those are going to end up being therapies for disorders that are very rare. So we have to start thinking about how we're going to have a regulatory approach that addresses this. So I'm going to focus now on advancing individualized gene therapy and think about some of the concepts here. Just to give you an idea of where we are right now, we have one FDA-approved systemic directly administered gene therapy on asemnogene, which is Zolgensma. This is for spinal muscular atrophy type 1. This is a disorder that spinal muscular atrophy type 1 is a disorder which is actually one of the, it's uncommon, but it's still more common than many other disorders. It's about an incidence of about 1 in 20,000 for type 1 of live births. It's unfortunately associated with death pretty uniformly by about age 2. The children become floppy infants and eventually require ventilatory support and eventually die and succumb to the disease. So this is a natural history, reasonably well understood. It led to the development of a gene therapy, which was approved. It was determined to be safe and effective. By the way, just again, as reference, I apologize, I didn't mean to be flipped before. That kind of caught me off guard. But we continue to stand behind the safety and effectiveness of Zolgensma or onasemnogene. And this was a therapy where the graphic that you see on the left side here shows that the children that were treated with this had a remarkable path where they did not require ventilatory support. They met developmental milestones and on a scale that had been developed at Children's Hospital of Philadelphia really showed a remarkable improvement. And the child on the right that you see running around there, that child was actually, the reason why I can show you her picture, she was running around on Capitol Hill by the HELP committee, one of the authorizing committees for FDA, or oversight committees for FDA, sorry. You can see that this is a child now at about age 3 running around. You don't need too many of those children running around like that to know that you have had an effect. This is a great example of when you have success by rational design. The clinical trial sizes don't have to be very large. We're not trying to put statisticians out of business. We're still going to need them for real-world evidence to work through. In this case, your 5th grader probably could have figured out that this therapy is probably working. Where does that leave us, though? So spinal muscular atrophy is in that sweet spot of disorders that occur in about somewhere over 100 to perhaps 10,000 people, where we can manufacture gene therapies and where they are commercially viable. There is enough money. We have to be realistic. There's enough money to be made that people want to go into these areas. Now, I'm just going to, for a moment, move from the grande to the venti size. The issue is if one of you out there in the audience said, look, we have a wonderful antisense that's in an AV vector that's modified so it doesn't really cause an immune response, and by the way, we're going to put a PCSK9 antisense oligonucleotide in there, and it's going to drop down cholesterol and LDL levels in people who have had heart attacks, and it's going to drop it down to, by 50 to 75%, and it's going to improve mortality. Wow, that'd be great. It would have a market size of tens of thousands of people. The problem is, we can do that today. You just can't make it. You just couldn't make the amount of product that you need. And so the scale up right now is something that's very challenging. And that technology, though, I suspect industry will find ways forward. People I know in this area are working, this area of the country are working on this, but the issue there is technology development to be able to scale up gene therapy production, because when you're making viruses, what do they like to do? They like to kill cells, and you're making them where? In cells. So this is going to be something that will be an interesting scale-up issue, and whether it will be better cell lines, non-viral vectors, or whatnot, that'll be interesting. At the other extreme, though, is the one I'm going to focus the remainder of my remarks on, the tall size. And you see I have a career in a Starbucks coming pretty soon when they throw me out of FDA. So the tall size, what about these gene therapies for 1 to 100 patients? Well, you want them made with high quality. You want them to be safe and effective. The problem is they're not going to be commercially viable, and we actually already have a case study of that. In Europe, there was a gene therapy that actually got conditional approval for lipoprotein lipase deficiency. It was a product that simply you just couldn't keep it, you couldn't make enough money off of it to keep it commercially viable. So at this point in 2020, we have this issue of being able to address genetic diseases with gene therapies, but not having commercial viability. Now, I suspect that this is going to be the case for the next maybe 5, 10 years, maybe 10 years, until we get to either synthetic biology advances or essentially technology advances in devices to automatically make various gene therapies. But right now, we have an issue here. And so we really, at FDA, have recognized that, and we have a vision for trying to facilitate the development of individualized gene therapies, particularly around adeno-associated virus. That's the vector that's facilitating direct administration right now the most. That's not because we love it, it's just because it's the one that's most advanced. There are other vectors in development. And we're working with our colleagues at NIH and others to help develop a public-private partnership to provide end-to-end solutions for overcoming key issues limiting the development and application of gene therapy in this area. What's really interesting, and this is one of the things we can do at FDA that actually NIH doesn't necessarily do, CDC doesn't necessarily do, CMS isn't into doing, we can see from end to end, from the patient need on out to getting a product developed and the delivery of the therapy to those patients. And one of the issues here that delivery is not underestimated here. We can't just have them studied in this population. Ultimately, if something is effective, somebody's going to need to keep making that available. And so that's why we think we need to have some public-private consortium, not just to get it through clinical development, but then to keep it available. Ultimately, we think it's not just going to help patients with those specific rare diseases addressed by such a consortium, but I think if we can actually address the need for the technical developments to make this happen, will advance the entire field of gene therapy. So when you think about it, what's the problem right now with individualized therapies? And this goes back to this great question about shouldn't more of this be done by academics? Well, it would be great if it could be. The problem is that academic investigators might want to develop a gene therapy because they have a patient population with a given disease, but they don't have experience with AAV. It's also hard to do investigation of new drug applications for phase 1 trials if you've never done them before. It's hard once you've actually done your trial. How are you going to make sure there's continued production of gene therapies for very small populations? And there's the cost and complexity of the regulatory process, the applications that need to be taken care of. And then how do you deal with a sustainable solution here if you're going to treat only 10 to 100 patients annually? And there are various potential solutions here which really go along the lines of trying to streamline by using a suite of vectors that are not proprietary, to using templates for protocols, to using templates for IND submissions, and to leveraging data in submissions, and potentially then to having a public-private partnership that could actually help make these and possibly even be sustainable through out-licensing of certain candidates. Because one thing we have learned about rare diseases is that some things that start to be very rare, we originally think they're very rare, they then sometimes turn out to be less rare. And so things that might start out as being commercially non-viable could turn out to be commercially viable, particularly if the regulatory process were de-risked. So the reason of our interest here is that right now it's taking people about 48 years to get through an investigator-driven process to get a treatment to patients. You have a therapeutic concept. It goes through development of making the construct, producing the viral vector, making GMP batches, doing the requisite toxicology studies, getting through the IND submission process, treating the patients, and then assessing the outcomes. Whereas if you had a consortium that was doing this, or a public-private partnership, you could leverage things like having vectors that were already qualified in terms of a lot of the preclinical work and the manufacturing nature information that's needed. And that could actually shave years off of that. And so you could get to patients sooner, we hope. And by cutting that time, hopefully in half, you'd make a real difference. And if you have a 2-year-old that's got a rare genetic disease who's likely to die by the time they're 4 or 5, that may make all the difference in the world. So we think that this type of a public-private partnership could make the technology more accessible to academic innovators, make this technology more applicable to a broader range of diseases. It would streamline the preclinical and manufacturing information needed for investigation and potential licensure. And it would facilitate the development of manufacturing and other advances that will ultimately benefit the entire field. But most importantly, it will bring therapies to individuals in need sooner. And it will bring therapies that we at FDA feel meet our standards for safety and effectiveness and quality. Because if anyone saw the New York Times today, we at FDA get hit on both ends. Either we're doing it too fast or we're doing it too slow. We're never doing it just right. And the more I stand here, the more I realize that something that someone told me shortly after I came to government is exactly true. If everyone hates you, you know you're in the sweet spot. Okay. So what's the next step for bespoke therapies? We are working with the FDA. We're at the FDA with the National Center for Advancing Translational Scientists and FNIH. We're working towards the potential development of a public-private partnership in this area. We are holding a workshop on March 3rd of this year to discuss the larger group of individualized therapies and how we might facilitate things. And I'll tell you a little bit more about some of the things we're thinking in a moment. We're really trying to do the additional work now up front in order to develop the regulatory framework necessary for these within the agency. And so what would that look like? Well, some concepts that we have in development are how could you leverage preclinical and manufacturing data from one application to another here? And one of the concepts that we're working on is how you could have a reference product and then develop variant products leveraging information on file and focusing just on the distinguishing attributes of variant products. So what would that be? So maybe you'd have a genome editing construct using CRISPR-Cas9 that was studied in a reasonably large population. It might not be a giant population, but it would be large enough that we felt that there was a robust data set on its safety and effectiveness, particularly focusing on the safety aspect, and we'd have the right manufacturing data on file. Well, so then you shift just the guide RNA in there, and then rather than submitting everything new, the idea would be you would submit the information that's relevant to the difference, which would be the off-target effects and the analysis that you'd need there to convince us that we're not going to have untoward effects. And that could help us really work through many more diseases more quickly. There are a number of things that have to be worked out and worked through here, but this is just kind of concepts in development. Another issue that comes up is you could imagine that someone might not find it commercially viable if there were only 10 or 15 patients treated a year, if they had to go through all of the development. and a development program. But if you had products which had been de-risked because they had been shown to be safe and effective, people might collect them. You could imagine that it would be a little bit like the generics industry. Maybe they wouldn't be huge markets, but there would be a living to be had by collecting them. And one way to get there is, how do we get products that are going to treat small numbers of patients across the finish line to a determination of safety and effectiveness? And although I'm not going to tell you I have all the solutions today, one way is to start to think about continuous reassessment of clinical data that comes in. So you have an endpoint that you're looking at. You know the natural history of the disease. You treat one patient. Well, you know that one patient's responded. And then you have some prior probability going into treating the next patient, and so on and so on. And at a certain point, though, you probably cross a line where, at least in the eyes of FDA, from the regulatory perspective, you have crossed into the realm of having demonstrated, reasonably demonstrated safety and effectiveness. So that's another type of idea that we're thinking about. So really what I wanted to try to tell you about is that we're really committed to advancing the development of products and novel therapies for populations of all sizes. I mainly focused on these small populations because that's really topical at the moment. We are working to help individualize product development. We have meeting types where people can come in early, so we can have early conversations. They're non-binding meetings, such as our Interact meetings at the Center for Biologics, to get information to academics and small sponsors about how they can move product development forward. We're trying to help people think about innovative clinical trial designs. We're happy to try to provide input and collaboration on novel endpoints. And we also, something I would love to say more about, but won't right now, is working to overcome limitations in manufacturing. So I'll stop there, and I think I have a couple minutes for questions.
Peter, so, with the NASMA gene, there were some issues with regard to data integrity, and then there was a media storm related to that. And you came out publicly and said, yes, things were done wrong, but it wouldn't have made a difference. This drug still belongs on the market. Is that a fair?
That's an absolutely fair statement.
Yeah, so the question I have is, what is the responsibility of the FDA to sort of temper these media storms when they, because the companies are often judged to be guilty and then be forced to prove themselves innocent. In this case, there were improprieties, but should others be taking this type of approach at the FDA to sort of bail out these people in these types of situations where the media response is perhaps greater than it should be?
It's very challenging, right? Because when we have an ongoing investigation, we really can't comment on it. And oftentimes it's because we don't know the extent of things. Sometimes we do. And the media tends to go where it wants to go with things. I think particularly, it was particularly, you never want to be the tastiest target. When you have a therapy that costs $2.175, is that it? $2.175 million, but which is still brings tremendous value to, if you think about it, I don't know, I'm just thinking about it from a parent's perspective for that child that you saw that picture, that's cheap at the price to have that child running around, they might say. Although from a public health perspective, you could come up with a different analysis. That was a very tasty target for the press to say, look at this. I hope, I don't know whether there are others outside of FDA that can help with this. I think we try at FDA, we tried to be very measured in what we said about this. I think, frankly, we did have to be really cautious here to make sure that we did put out some firm statements about not wanting to have people take shortcuts with data. But it was sad how much of a firestorm there was about that, because I had to spend a lot of time actually calming down the patient population, that with this, who were suddenly panicked that this product was not safe or not effective. And we would, at this case, were very comfortable saying there was no issues with that. This was not a safety or efficacy issue. And I'll repeat something that I probably shouldn't have said at the time, but since I'm quoted in the New York Times as saying it, I can repeat it now, which was that, this is one of those cases where, boy, somebody did something really stupid, because if they would have left well enough alone, the product still would have been likely have been approved. Sure.
Peter, I had a question. Art Chochala, Regulatory Novartis. My question is, has the agency considered perhaps what I'll use the word a conditional approval? In Europe, it's certainly a process that's been ongoing here. Would you use the example of this dyslipidemic patient population, small number of patients? Would you have considered, or have you discussed perhaps accepting a clinical trial of N of 1, if you will, allowing the drug only to be used in restricted patient population, restricted physicians, restricted distribution, and so on? Would that be something the agency might consider or has considered?
We've thought about conditional approvals a lot, and I think we like to believe that our current accelerated approval process and our current processes suit us pretty well in this area. I think at the end of the day, the conditional approval process works well in certain places because of the different nature of how therapies are paid for. I would really hate to have an approval process that didn't lead to robust enough data that insurers in general would accept the data that are saying, so that I would rather find a way that we can get to some full approval, or accelerated approval, or full approval that insurers will reimburse for these products, than have a conditional approval where some insurers might say, look, we don't really have enough information here, so we'd have haves and have-not. So we've given this a lot of thought, and I think you'll hear more probably about whether conditional approvals are reasonable. I think we have access pathways for this, so I think we have ways to have patients have access through the IND process, and I think that that's the way to get through here, hopefully through small trials that will eventually get things to market, rather than getting to some point where, what the amount of data that you might need for a conditional approval versus a traditional approval or an accelerated approval, it might not be all that much different, and it's probably better to have products that insurers will feel comfortable reimbursing.
Great, thank you.
Okay, I'm going to use my prerogative, since I have a microphone right in front of me, to ask you a question. Look, Al Sandrock and I were involved in the approval of Spinraza, right, which is also for SMA. And so we know firsthand what the impact is on those families. So it would be a tragedy if that 3-year-old running around got to be 8 and suffered some irreparable nerve damage because the effect starts to wear off. And so the hope is it's a one-time treatment and it lasts a lifetime. We don't have those data yet. So are you doing anything to monitor this over time, and how do you view that?
Yeah, very, very certainly so. In fact, there is a long-term follow-up draft guidance that's out there. We are asking sponsors of gene therapies to follow patients for a risk-based approach, anything from 5 years to 15 years. And I think we all are worried about exactly what you're worried about. The only good piece of news is that there are different AAV vectors that may be able to help here with redosing in the future, and people are developing essentially AAV vectors that may be able to evade antibody formation somewhat. And all that failing, there are also ways to try to deal with desensitization. But your point's well taken, and that's why patients are going to need to be followed to make sure we understand how durable are these responses. That's going to be ultimately probably more so than even in SMA. For hemophilia, that's going to be a key issue as we go along, especially there, because we're talking about years and years where we have a very safe and effective alternative around right now.
Okay, last question.
Hi, Peter.